going, why do we have to learn this? Okay, but it is what it is. All right, so here's where we're at now. Is that we spent last week going through, last couple of weeks actually, going through a series of looking at how components in RTK signaling pathways can become converted to oncogenes and give rise to abnormal cellular proliferation, okay? And you can see that you can basically generate oncogenes pretty much through the entire pathway, starting all the way at the extracellular portion of the RTK receptors to working all the way down to the nucleus, to the transcription factors that actually activate cyclin D, cyclin E, and basically getting the cell cycle underway. So multiple opportunities in these pathways for the acquisition of oncogenes, okay? But the question you have to ask then is, well, where did these come from? What caused these particular mutations to arise in the first place? I mean, did they just happen spontaneously out of the blue? Are they relatively rare? Are they intrinsic, or are they controlled by extrinsic or external factors? And so that's what we're moving into this next section of looking at cancer. And it's really focusing on what you might consider to be the genetic basis of cancer, all right? And the way to think about it is the following. Think of cancer as a phenotype, okay? Now, it's a broad phenotype, and it's always changing. It's always evolving, okay? But the point is, cells that become cancerous to a certain extent will continue to inherit that cancerous phenotype. That's going to change, but it's inheritable. And very, very rarely do cancer cells ever revert or go back to a normal phenotype. Very, very rarely. And so that right there, the fact that cancer is heritable, is basically saying there's a genetic component, okay? So that means there have to be alterations that take place at the DNA level in order for these to be propagated every time cells divide, okay? So one of the things that we went through is proto-oncogenes get converted to oncogenes by what you would consider to be dominant active mutations, okay? These are mutations that are causing a signaling component to propagate a pro-proliferative signal, even in the absence of any extracellular growth factors, okay? Conversely, what you're going to see is that most tumor suppressors are going to be loss of function, which means they're going to be recessive mutations. Now, some of them can also be dominant negatives, and we'll talk about some of those. But generally, proto-oncogene to oncogene means you gain a function. Tumor suppressors are correlated with a loss of function, all right? So, what causes them to take place? The first thing is that we talked about a number of different types of mutational events. So the first thing to appreciate is mutations aren't just changes in individual nucleotides of DNA. You already saw examples of where you can have deletions, you can have translocations, you can have gene fusions. So you're looking at a spectrum of DNA damage that can generate the mutations that are responsible for converting proto-oncogenes to oncogenes and losing tumor suppressors, all right? So there's not one single predominant type of mutation, all right? So you've got DNA damage, first and foremost, as the causative event. So then you go, well, what causes the damage? What are the sources of the damage? And what you're going to see is that with a very, very small exception of a small number of inherited cancers, genetic cancers, most of them are environmental. And in fact, one way to think about cancer is that it's an environmental disease, all right? And that is, what you're going to see is that the causes of DNA damage are extrinsic. And basically what it comes down to is exposing your cells over your entire lifetime to different types of DNA damage that become cumulative in their effects. And in essence, this also ties in with the fact that the reason cancer is more prevalent now than it was 50, 100, 200 years ago is simply because we survive longer. When you take out infectious diseases, you add the fact that you have childhood immunizations, the development of antibiotics to ward off bacterial infections. What it now means is it simply put, we live long enough now to where our bodies are exposed to more of this damage. And the effects are cumulative, and in essence, think of your genome as a big target for mutational events. And at some point, the cumulative effects of this damage will overcome or circumvent the repair mechanisms and the checkpoints that our cells have to keep these things from propagating. Okay? So in essence, it ties down with the fact, again, cancer is progressive, takes a very, very long time, and it's basically due to multiple damaging events that are going to occur in our genome. And what it is, it's a process of adaptive mutations that are being selected for. Okay? So cancer cells are being selected for their ability to survive. So the types of mutations that they're undergoing are adaptive to the extent that most of the mutations that happen in cancer cells don't confer any selective advantage, and most cells die. They don't propagate. You're looking for outliers at a very, very low frequency that a particular DNA damage event or a mutation allows them to survive a particular selective pressure that's being acted on the cells at a particular time. And as you're going to see, that's also always changing. It might be hypoxia at one time. It might be the immune system at another point. So the simple fact is, the cells are undergoing Darwinian selective pressures to acquire mutations that allow them to evade or bypass whatever pressure is being put on them at that time. And so the term that we're going to use to describe this phenomenon is up there. It's called clonal expansion. And that is, we're going to look at, over the course of time, you'll have a small cohort of cells that undergo an adaptive mutation. They out-proliferate their neighbors, okay, simply because they can. Well, then a selective pressure comes in, it changes, and now what it is, the process starts over again. And so now what you're doing is you're taking this first cohort of cells, they're subjected to pressure, and out of that you now accumulate or acquire another generation of cells that have now acquired additional mutations. All right? And what you're going to see is that by the end of the process, when you look at a cancer cell, a full-blown metastatic colon cancer cell, metastatic breast cancer cell, and compare its genome to the normal cell that it originated from, the cell hasn't acquired a handful of mutations, a dozen mutations. They've acquired hundreds of mutations. And the challenge is in sorting out which of these mutations are what are called the drivers, the ones that are actually responsible for causing the cell to abnormally proliferate, and which mutations are called passenger. Passenger mutations are going to be the ones, they just happen, they get carried along with the driver mutations, but it's very, very difficult teasing out, again, trying to understand at a particular time, which mutations are actually responsible for that cell's proliferation at that time, and which ones just happen to have occurred, and they're manifested in the phenotype of the cell at that time. Okay? Now, that's the bad news. The good news is the following. We've got remarkable enzymes that can repair just about any type of DNA damage. Double-stranded breaks, mismatch, ultraviolet irradiation, you name the type of damage, we've got enzymes that can fix it. And most of the time, they do. The Achilles heel in the process is that those repair enzymes are normally not expressed. They're silenced because we don't need them. They have to be induced or switched on when the damage takes place. And here's where the weakness in the system comes into play. And that is we've got predominantly one tumor suppressor, more than all of the others, that has the responsibility for activating these repair enzymes, and that's the p53 tumor suppressor. We've got lots of tumor suppressors. Some of them are redundant with p53, but none of them have the capacity to function in as many cells as p53 does. And they also don't have the capacity to do everything that p53 can do. That's the weak point. Why do we put something so important under the control of a single gene making a single protein? Because of all the tumor suppressors we have, p53 is the only one that can immediately stop the cell cycle at any point during the cell cycle progression. So basically halt the cell from continuing through a mitotic cell cycle to give it time to fix the damage. So that way the damage isn't inherited and propagated in the daughter cells. Second point is p53 also has the job of activating the DNA repair enzymes itself. It's a transcription factor, it's a DNA binding protein, so it also turns on the expression of the repair enzymes. And then finally, p53 also holds the life or death decision over our cells, because only p53 has the ability to cause cells that, if they were unable to fix the damage, cannot get out of the cell cycle arrest it imposed. p53 is the only protein that can cause a cell to enter apoptosis and kill itself. Okay? So you're going to see that p53 is the linchpin for all of these things to happen. And again, normally it works quite well. But when it doesn't, things can very, very quickly go awry. And because of this, what you're going to see is that there's a lot of attention that's being focused now on trying to restore p53 functionality in tumor cells. The gene is still there, the protein is still there, but what you guys are going to see is most of the mutations that inactivate p53 function do so by destabilizing the protein. So if you can just increase its stability to where the protein can accumulate, then you've got the possibility of having a therapeutic option there that will allow the cell to basically kill itself if we can restore the functionality of this one protein. Okay, so the environmental connection with cancer goes way back, okay? So you can go way, way, way back, even into the 18th, 19th century. And it was observed that there are certain types of cancers that have, shall we say, an occupational predisposition. Okay? Chimney sweeps, especially in England back in the 17th, 18th, and into the 19th century, basically the way that you clean chimneys back then is you send a little guy or a little kid climbing down through the chimney. And they would carry a brush and scrub it as they went down through it. All right? They would be covered with these coal deposits, which are highly carcinogenic. And so these individuals, for example, came down with scrotal cancer at a much, much, much higher frequency than anybody else. Watchmakers came down with bone cancer at a much higher frequency because back then they used the radioactive element radium to basically make points on watches that would glow in the dark. It's how Madame Curie died. It's because of all of her work with radium and uranium, she basically died of radiation poisoning. Rosalind Franklin developed ovarian cancer because of being exposed to X-rays with minimal shielding back in the 1950s. So there's always been this observation that certain types of environmental factors, okay, and you can see here's a list of them here, that are associated with certain types of cancer. All right? And as you go up here, if you take a look even at the types of cancers that are out there now, you notice that the ones that are most prevalent have a discrete non-environmental connection. For example, as we're going to see, tobacco-related, smoking. As many as a third of all cancers are associated with that. About another third are associated with lifestyle or dietary contributions. And so this is the one that we'll have you thinking about as you just went through the Thanksgiving holiday. Okay. Oh yeah. By the way, here's the first experimental case of where an environmental agent was directly used to cause cancer. You can never get away with this experiment now. PETA would be after you big time. In 1915, a Japanese oncologist, Yamajiwa, basically took different carcinogenic compounds or suspected carcinogens and painted them on the ears of rabbits. All right, this is one that was preserved. It's in a nice jar. And what he did is by painting the ears with these different coal tars in this particular instance, they developed these very, very large tumors. So that was the first experimental evidence that you could cause cancer by treating normal cells with an external agent that was suspected of causing cancer. All right? But it took until the late 1970s and the early 1980s for a series of experiments that were done by a scientist named Bruce Ames. And Ames was basically interested in making the connection between mutagens and carcinogens. All right? The two words are not synonymous. A mutagen is a compound that damages DNA. And as you're going to see, it can do it directly, meaning the compound can directly, upon exposing your DNA to it, can directly cause the damage, or they can be indirect. And an indirect mutagen, or an indirect carcinogen, as you're going to see, is a compound that's initially inert, but when you ingest it, breathe it, digest it, you metabolically convert it to a compound that can now become mutagenic or carcinogenic. So Ames wanted to establish the relationship between these two. And so what Ames designed, he was a microbiologist by trade, and so what he wanted to come up with was a very simple assay or a simple screen that could be used to identify mutagens. Okay? And it became known as the Ames test. Okay? So we're having to step out of cancer here for a moment and instead move into bacteria. All right? The strain of bacteria that Ames used was a strain of Salmonella. Same type of bacteria that causes food poisoning. All right? What he had done is he went into this strain of Salmonella and genetically engineered it so that it could no longer synthesize the amino acid histidine. Okay? So he took the gene, this one gene that was required for histidine biosynthesis, and introduced one specific mutation, okay, a point mutation, that inactivated that gene. So now what it means is the starting strain of Salmonella that he's used is going to be described as a His oxotroph. Oxo meaning it needs help in order to grow. Trophic meaning growth. Oxo meaning help. It needs help to grow. So the original strain of Salmonella that he's using, we refer to it as a His oxotroph. Sounds kind of cool. What it means is the only way then that that strain can grow in the absence of histidine is if it undergoes a mutation. And the mutation has to be very precise. It has to be a mutation that is so specific that it reverts the original mutation that he put into the histidine biosynthetic gene. He's making a very, very specific target for a mutagen. Okay? What it means is this, is that you're going to use as your assay the conversion of a His oxotroph to what was now be called a His prototroph. Prototrophic means it can make its own histidine now. All right? It's very selective. So what it means is this. If you were to take Salmonella and treat them with a very, very weak mutagen, okay, that compound is not going to cause mutations at a high enough frequency to go in and basically hit the specific target that he set up to convert it from a His auxotroph to a His prototroph. A potent mutagen, something that is very, very, very mutagenic, at a very low concentration, is going to cause tons of mutations, okay? And what it means is that at a much higher frequency then, a potent mutagen will cause the conversion of His auxotrophs to prototrophs at a much lower concentration because it is so much more mutagenic. Okay? Now, you might be thinking, well, why didn't he do the opposite? Why didn't he just start with a strain of Salmonella that was a prototroph, and now why don't you just look for mutations that cause it to become an auxotroph? That is, mutations that would not allow the cell to grow in the absence of histidine. Why do you think he didn't do that? It has to do with the stringency. Okay, think about it. If you basically start with a cell that's a prototroph, okay, it can make its own histidine. Is it all that difficult, can you imagine, to go into one of the biosynthetic genes for histidine and acquire mutations that will knock it out? No. So it's not very stringent. It would be very, very easy to convert a strain that was a prototroph to an auxotroph by mutational events. That means even a weak mutagen would be able to do that at a relatively high frequency. You'd have a lot of background. That's why he chose this screen. It is very, very high stringency. Okay? But the other thing that Ames does in this assay is he also takes into consideration the possibility that compounds may not be directly mutagenic if you treated cells with them directly. He also took into account that certain compounds may be intrinsically non-mutagenic, but after they have been metabolized, they will become potent mutagens. Okay? So let's go through the screen, and you can see what he did. Okay? Let's just start here looking at direct-acting carcinogens. Okay? So what Ames literally did is he went through and had his students literally take everything they could get their hands on and take it off the shelf. Everything. As you're going to see in a minute, when I say everything, I mean everything. Take anything you can imagine that you suspect as being a carcinogen or a mutagen. Now, some of them are no-brainers. Some of them you might have to think a little bit harder about. So what he did is he took these compounds here, and what you could do is you just directly took your compounds and added them to Salmonella auxotrophs. And you're doing it at different concentrations. So you're doing a dose-dependent conversion of His auxotrophs to prototrophs. And all you're doing is that you're measuring how much of the compound do I have to add in order to generate, say, 100 Salmonella colonies here. Okay? So obviously, if something's a weak mutagen, you're going to have to use a much higher concentration of it than if something's a potent carcinogen or mutagen. Okay? And here's where he went back and didn't stop with just doing that experiment. Okay? Because obviously, if you did this, you would be missing compounds that are not mutagenic on their own, but would be if they were metabolized. So the enzymes that are generally responsible for converting metabolically activating carcinogens or mutagens are found in the liver. And these are enzymes that are basically called cytochrome P450 enzymes. Okay? And these liver enzymes, this is why the liver is the target for so many carcinogens and why hepatic or liver cancer can be so prevalent. The reason is that the liver, more than any other organ, especially when you consider your entire circulatory system, your blood goes through the liver. The liver has enzymes that can take compounds and metabolize them to become active carcinogens or mutagens. It doesn't do it to cause cancer. The cytochrome P450 enzymes are there to basically take hydrophobic compounds, convert them to become more water soluble so that they can be excreted. That's the purpose of having the enzymes. Okay? But here's what Ames did. The same compounds that he tested directly over here, he also took them, and what he did is he took preparations of rat liver, ground them up, made soluble enzyme extracts, and now he pre-incubated his test compounds with this liver extract. And you could substitute purified cytochrome P450 enzymes for the liver extract. And he would do that before he took the compounds and added them to the salmonella. So now you can see Ames is covered. He's got over here, these will allow him to identify compounds that are direct acting. Over here, now he can find compounds that are going to be referred to as pro-carcinogens. They don't do anything on their own. But after they've been metabolized, now they become what are called ultimate carcinogens. And sure enough, what he found is a significant number of the compounds that when you directly add them to salmonella don't cause mutations to occur. After they've been exposed to these cytochrome enzymes, they become highly mutagenic. So in fact, the vast majority of compounds that are capable of damaging DNA and ultimately causing cancer, the majority of those compounds are in fact in this group over here. They're not direct acting, the majority are indirect acting. So this takes care of the mutagen part. But he also then took care of the carcinogen part. So here's what he did, is that on the x-axis down here, okay, here's a log scale plot of looking at the concentration in micrograms of these compounds that it takes to generate 100 salmonella revertants. Okay? That's the assay that we just went through. So obviously, if you require a lower concentration of the compound to generate 100 colonies, as opposed to 10 to the second over here, if you're towards this end of the graph, you're more mutagenic. Okay? He then took these same compounds and then treated mice with them to basically look at the induction of tumors over a 2-year period. And he's also doing this in a dose-response fashion. So we basically have 2 log plots that shows a series of the compounds that he tested. And that's the correlation that he found. You couldn't ask for a more direct correlative result than what Ames came up with. And what he found is that there's an absolute invariant direct correlation between mutagenicity, meaning the ability to cause mutations, and carcinogenicity, the ability to cause cancer. Absolute perfect correlation. Now, this doesn't say if the compound's direct or indirect acting, but it does say that basically the more potent a compound is in its ability to damage your DNA, the more potent it is in terms of being able to cause cancer. All right? So Ames went through and did this systematically over about a 10-year period and categorized an entire collection or a directory of carcinogens that were out there that people are exposed to every day. What this keyed into, though, is that the person who saw this data and then used it immediately to start trying to isolate oncogenes himself was Robert Weinberg. So Weinberg was the first scientist, as you're going to see, was the first scientist to actually clone an oncogene. But the reason he was able to do this was premised on the Ames result. Because what this told Weinberg was the following, It means that if I look in a cancer cell, at least one gene has been genetically altered. It's different than the same gene was in a normal cell. And what it meant was you should be able to functionally isolate that gene and determine what was the mutation that caused its conversion from a proto-oncogene to an oncogene. But the seminal idea that he got to do those experiments came from the Ames results here. And so as you're going to see, the Ames test built directly provided the foundation that Weinberg and other investigators are going to use to actually clone oncogenes from a variety of human tumors. Okay, so here's the fun part. You want to know what he found? Okay, this is just a small compendium of all the stuff that proved to be carcinogenic in the Ames test. Okay? One of the things that everybody gets annoyed with or fed up with are people that don't eat meat products, and they think they're such purists, and they think they're so healthy, and that everything's organic, and nothing bad's going to happen to us. Well, this ought to dissuade you of that. So if you're a vegan, you have no moral superiority when it says your diet is going to cause you to have a less of an opportunity to get cancer than us carnivores. No. There it is right there in blue and black. This is everything that Ames found. They call it his roster of carcinogens. Okay? So if you take a look, these are some of the more potent compounds that he came up with that are basically, they proved to be carcinogenic in the Ames test. Black pepper, common mushroom, celery, rhubarb, cocoa, mustard horseradish, alfalfa, that's a good one, alfalfa sprouts. You gotta love this one, burnt materials. This actually covers everything. On average, several grams of burnt material are consumed daily in the form of bread crust, burnt toast, burnt surfaces of meat. So there you go. Take a look here. These are also compounds that you can find in everything else. So, okay. So you look at this slide and you go, is there anything out there that does not cause cancer? Probably not. Your only hope would probably be to drink distilled water. But don't do it in those plastic bottles that you have, because the plastic's probably carcinogenic, and it leaches into the water. In fact, there was a whole, remember the camelbacks that came out a few years ago? They got rid of them because they had this really nasty carcinogen in the plastic that they quit making them out of, but they only discovered it after several years. So the bottom line is this. You look at this data, and I remember hearing Ames give a talk about this, and everybody was just sitting there thinking, there's no escaping it. We're doomed. It's inevitable. You're going to get cancer. And the answer is no. Most of the time, we don't. Because most of the time, we've got these spectacular DNA repair enzymes to fix the damage that these things do. And most of us, most of the time, have functional p53 genes that can activate them and basically cause the cancer from ever developing in the first place. The other thing to be mindful of is that we have very robust immune systems. And it's immune surveillance that actually captures most cancer cells very, very early on and destroys them at the outset. So the bottom line is that we are very, very well equipped to circumvent the damage to our DNA that you're going to be exposed to on a very regular basis. And when you go back and you look at everything that you typically put into a Thanksgiving meal, you'll find it up there. Anyway, just something to keep in mind. Okay, but let's focus on one type of cancer or carcinogen that you might think we do have a means of controlling, and that's smoking. Okay? What this table shows over a time period going back over 100 years is that you're looking at per capita consumption of cigarettes. And smoking and cigarettes has probably the single most direct environmental correlation with a particular type of cancer than any other carcinogen out there. Okay? Here's what's interesting, is that if you take a look at the line in black here, so start off with this. If you start taking a look at per capita cigarette consumption, notice it kind of goes through a couple of spikes. It's almost bimodal here. It kind of comes up to where it's relatively low here, and then you notice that it starts going up noticeably during the 1920s. Okay? And that's just because culturally and socially, smoking became very, very acceptable. It became very hip. It became very cool during the 1920s. So you start seeing an increase in consumption of cigarettes during that time, the Roaring 20s. Dips a little bit during the Great Depression. But then it really starts spiking upwards again as we go into the 1940s. The reason for that is that during the Second World War, all of the troops in the armed forces were issued cigarettes as part of their basic kits. And so cigarette smoking skyrockets during that time, during the war, and people are getting hooked on it because you're dealing with a lot of people that are 18, 20 years old that are basically in the armed forces, and now they're smoking at a very, very high rate. Okay? And so what happens is that you notice it starts going up, and then about here, in the 1950s, in particular moving into the 1960s, notice that per capita consumption of cigarettes starts dropping back down. That's because that shows you the accumulated time that it takes for all of these people that started smoking back here in the 20s and 40s. This is where you notice that the incidence of cancer death rate skyrockets. So these are predominantly men, okay, because smoking culturally had sort of a sexist component to it up until the 1960s. And the reason is that smoking did not become fashionable or socially acceptable for women until the 60s. So there's a lag between when you saw cancer deaths for women starting to come up from when you saw it starting to peak with men. Okay? And that's simply because men were smoking more commonly and more frequently for about 20 to 30 years before it became fashionable in women. And so that's why you notice here, you can see this difference in the onset of cancer deaths between men and women, and it's just basically due to that simple demographic observation. Okay? You notice that at no time that the number of women smokers ever exceeded the number of men. Okay? So men were always smoking at a much higher frequency, and because of that, most of them encountered lung cancer and died, typically in their 50s and 60s. Okay? So that's the demographics of smoking. Here's what it actually does. The culprit, as many of you know for cigarette smoke, is an aromatic compound, benzopyrene, over here. Okay? You can see it's aromatic. It's also extremely hydrophobic. Okay? The problem with benzopyrene is it's relatively water insoluble. This is one of those indirect carcinogens. It gets very, very actively metabolized by cytochrome P450 enzymes, both in the liver and in the lung. And what it does is that the modification that's being done by the cytochrome P450 enzymes is that what you're doing here, in order to make it more water soluble, is you're basically generating a highly reactive epoxide over here on this ring over here. And then the epoxide ultimately gets converted to hydroxyl groups that make it more water soluble so it can be excreted. That's why you're doing it. But the problem is it's these highly reactive epoxides where we can generate one down here. You can also generate one up here on this particular ring. And this epoxide is highly reactive with the base guanine. And what it does is that they're only showing you here the terminal rings. But what it does is it forms this covalent adduct on guanine. And enzymatically, we can't break that. And what it does is by sticking benzopyrene and forming this covalent adduct with guanine, is that we distort the structure of that modified guanine. So as you know, guanine normally base pairs with cytosine. It makes a GC base pair. Here's the problem. If you don't repair or remove that modified base, we have enzymes that can remove the modified base and replace it with normal guanine. Everything's good. But if that enzyme fails to work and DNA replication takes place, the two strands are going to separate. So the strand on the opposite side of this modified G, we've got a C. So we're going to pick a guanine across that just like you normally would. The problem is when you're trying to replicate the strand that's got the modified G. Because what happens is that modified G no longer pairs with C. It pairs with T. So what happens after DNA replication is you're now in a situation where you have this modified G paired with T. Okay? So the question is, what's going to, I'm sorry, it's an A. Sorry, guys. Got my Watson crick screwed up. This modified G pairs with A. So now we've got a GA mismatch. Well, now what happens is the cell looks at that. The adenine looks normal. The modified guanine clearly isn't right. So what the cell will do is remove the modified guanine and replace it with T. So what happens after going through a couple of rounds of DNA replication, the modified G will get converted to T. Okay? And as you're going to see, if that mutation takes place in the coding region of the p53 gene, as you're going to see, the p53 gene has what are called mutational hotspots. There are certain nucleotides in the p53 gene that are, the Gs are very susceptible for this type of mutation. And that G to T transversion will basically inactivate the p53 gene. They're going to cause missense mutations that cause the p53 protein to be very unstable. And in fact, what you do is you find this in a third of all lung cancer smokers that develop lung cancer. So their lung cancer cells will have this exact signature in their p53 gene that will inactivate it. Okay? So here's some interesting statistics. This just came out in the Washington Post a couple of weeks ago. And it's a result of a study that was carried out by the CDC. And what they're looking at is following the decline in cigarette smokers over the last 10 years. So it went from 45 million smokers in 2005 to just under 37 million smokers in 2015. So here's what's interesting. Look at the breakdown on groups that had the largest decline in smoking based on these different demographics. Notice that the single largest group that exhibited the greatest decline in smoking, you guys. People under the age of 24, it went down by more than almost half. Okay? You notice as you get older, people are less and less inclined to stop smoking or to not smoke. So you notice that the most recalcitrant ones were up here, older people that have probably been lifelong smokers, so it's much, much harder for them to give it up. If you go down and you basically look at it based on race or ethnicity, you can basically see that Asians and Hispanics actually have the lead in terms of the populations that have the largest decline in smoking. But here's what's interesting, is if you take a look up here, the percentage drops are the elderly and people that don't have high school diplomas. So the interesting connection here is that what it is saying is the single most effective way to reduce lung cancer or diseases that are associated with cigarette smoking is going to be what? Education. So people that are informed, that know the risk, are going to be the ones that are most likely never to start smoking in the first place, or they're going to be the individuals most likely to give it up, because it can recognize the long-term hazards from this. Okay, So here's what we're going to get into then for Wednesday. This is the guy who does it all, P53. And the way that we need to think about this network is the following. We need a sensor, we need an enabler, and we need an executioner. The sensor has to be something in our cells that can detect DNA damage. And ideally, you want that sensor to work anytime in the cell cycle, because you never know when the DNA is going to be damaged. So our cells, first and foremost, have to be set up. So if we take a look at the cell cycle, we have to have a mechanism to sense when damage took place at any time. And we do. The sensor is going to be a stress-activated kinase. There are two that are interrelated, ATM and ATR. They differ only in the types of DNA damage that they can sense. Okay? They are part of a family of stress-activated protein kinases. They're serine-threonine kinases. These are kinases that live in your nucleus, and they are constantly moving up and down your DNA. So ATM and ATR are the sensors. They're constantly touching and feeling your DNA, and they're looking for damage. A double-stranded break, a UV crosslink, a GT mismatch. ATM and ATR sense that damage. And when they sense it, for example, by binding exposed single-stranded DNA, it activates their kinase activity. So these are DNA damage-activated kinases. And what they do is that when they are active, they're going to phosphorylate P53. They also are going to phosphorylate another pair of kinases over here called CHEK1 or CHEK2. They are checkpoint kinases. We'll put them on hold until Wednesday. But the key thing is this. The DNA damage sensors phosphorylate P53. And what you can see is that P53 can get activated pretty much throughout most of the cell cycle. But here's where the system has a bit of a flaw. And that is if we look at G0 through G1. Remember, where are most of our cells at any particular time? G0, which is the only part of the cell cycle where you can repair DNA damage before DNA replication takes place. G1. Can you guys see the weak point right away? We don't have any redundancy for activating DNA damage repair during G0 or G1 because the only pathway we have is P53 dependent. So P53 is the only way that we can activate DNA damage checkpoints during G0 or G1. And that just happens to be the part of the cell cycle where most of our cells exist at any particular time. So, where we'll pick it up on Wednesday then is what does phosphorylating P53 do to it? And then secondly, how does P53 go about executing the repair or the death program to fix it? Okay? Pick it up on Wednesday.